Hello and welcome to the next video for higher biology. This one is looking at cellular differentiation and is the fourth key area in your first unit, DNA and the genome. So before now we were looking at proteins and phenotypes, so how one protein can help change the phenotype of an individual organism. What we're going to be doing now is moving on to cellular differentiation, looking at how cells are different, before we move on to look at embryonic and tissue stem cells. So for today we're looking at understanding the need for different cell types. You need to be able to define the terms cellular differentiation, meristem and stem cell. You need to be able to identify the locations of meristems in plants and identify the locations of stem cells in animals. When we look at cellular differentiation, what we're referring to is the process by which a cell can express certain genes, allowing it to produce proteins that are characteristic for that type of cell. That both allows a cell to carry out specialised functions and allows different organisms, multicellular organisms, to be made up of lots of different types of cell, all of which are carrying out different roles and forming different structures and organ systems. The reason that all of these cells can differ is because the genes that are within them can be switched either on or off. So when a new cell is produced by mitosis, or two new cells to be more accurate, they are really non-specialised or undifferentiated. Specialisation is going to determine which genes are switched on and off and is going to give the cell a particular shape and a particular purpose in which it can carry out to support the organism. So for example, if this cell has formed two identical daughter cells, in this one it might express a specific neuron-specific gene and it will turn itself into a neuron cell. The other one, on the other hand, may express an epithelial-specific gene and as a result it will turn into an epithelial cell. So two very different cells arising from one individual single cell. Now, differentiated cells have a limit to the number of times that they are allowed to divide. So over time, they will become unable to continue to make new cells. So for this is our specialised cell here. At this point, this one could divide and form another neuron. But after a certain amount of time, there's only so many times the nucleus is capable of doing that. And that's one of the things that eventually causes old age. There are only two cell types which are able to replicate indefinitely, and they are stem cells or cancer cells. So we can see here we've got our stem cell which is carrying out cell division, and this stem cell could go on dividing and dividing and dividing and dividing and dividing, whereas the specialised cell, once it becomes specialised, it has a limited number of divisions within it. Before any cell is differentiated or specialised, it's called a stem cell. And stem cells are cells that can make more of themselves, so they can duplicate, so this stem cell could turn into two new stem cells, or they can become almost any type of specialised cells. So stem cells are involved in both growth and repair. So these stem cells can become capable of turning into any type of cell within the organism. In plants, stem cells are referred to as meristems, and the regions of meristems that are found tend to occur both at the tip of the root and the tip of the shoot. So up here, if we're looking at the shoot, so this is zoomed in from this little bit of the plant here, and right up at the top we've got a region of meristems. Similarly down here we've got a little zoomed in section of our root, and right down at the bottom we've got a little group of meristems. And what they're able to do is add length to the plant, so letting it grow up or down as appropriate. However, we also have lateral meristems in plants, and these occur at the sides of the stems, and that allows the stem to get thicker or fatter. In animals, stem cells can exist as either embryonic or tissue stem cells. Embryonic stem cells are found at the blastocyst stage of a newly fertilised egg. This is usually going to develop at around 5 to 8 days after fertilisation. So if had our fertilised egg is developed into a few-celled embryo, over time that bundle of cells starts taking on this particular form, and within it we have a group of embryonic stem cells. We also have tissue stem cells, are also referred to as adult tissue stem cells, and these are present in various different tissues throughout the human body. For example, the surface of the eye, the brain, skin, breast, intestines, testes, muscles, and bone marrow. And any one of these, usually at this point, adult tissue stem cells can't turn into any different type of cell. They can only really replenish the cells of the tissue to which they belong. But that's what we'll be looking at in our next lesson. So you should be able to define the term cellular differentiation, the process by which cells express certain genes to produce proteins for that cell. A meristem being the stem cell of a plant, and a stem cell being special types of undifferentiated cells within an organism. Locations of meristems in plants, either at the root tip or the shoot tip, and laterally in the sides of the stem. The location of stem cells in animals can be found either in the blastocyst stage of a fertilised embryo, or it can be found in various tissues throughout the human body, such as the skin, the brain, or the bone marrow. As ever, we'll be going over this again in class, and if you've got any questions, please make sure you ask them.